the second molecule. So that is your plane of symmetry. Okay. So if you mix these both enantiomers, we call it as a racemic mixture. Okay. So this all terminologies which I just give a brief about is they all come into this slide. So this is the only slide that if you are planning any drug molecules in the synthesis, everybody must know this. And this is important. Why? Because why asymmetric synthesis? Number one, often only one enantiomer is biological active. अब आपने enantiomer A कायरल सेंटर से आपके 2 enantiomer बनेंगे, 2 कायरल सेंटर से आपके 4 डिफरेंट मॉलिक्यूल्स बनेंगे. आपकी रिक्वायरमेंट तो सिर्फ एक है जो अच्छे से एक्टिविटी देगा, रेस्ट 3 आर गोइंग टू इंटरफेयर. सो often only one enantiomer is biological active. Now can you just feel it? अगर आपका सेकेंड enantiomer भी पार्ट हुआ, क्या ड्रास्टिकल चेंज लेके आ सकता है? कैन यू रियलाइज द थैलिमिडो केस वेयर प्रेजेंस ऑफ ओनली वन enantiomer can lead to the deformities. So that's why only one enantiomer is biological active. What is the second question? Both enantiomer may have a different activity. I have prepared a drug molecule, I get 2 enantiomers. One enantiomer is antibacterial in nature, other enantiomer is antiviral in nature, anti-inflammatory, anti-cancer, anything, because in 3 dimension they are interacting with the enzymes in a different way. Okay, so both enantiomers may have different activity. Third query क्या हो सकती है? One may be desired, the other is inactive and undesired. आपको एक enantiomer चाहिए, दूसरा नहीं चाहिए, वो अनडिजायर्ड हो सकता है. जो अनडिजायर्ड enantiomer है that can lead to the environmental pollution. So if you see, sometimes optical active enantiomer is twice as active as compared to racemic mixture. अगर आप यहाँ पे स्क्रीन में एक एग्जाम्पल देखोगे, इस एग्जाम्पल में आपके पास सिंपल एलिफेटिक चेन है और एक 5 मेंबर रिंग है. नाउ देयर आर 2 इंपॉर्टेंट एस्पेक्ट इन दिस. डबल बॉन्ड है. अगर आपके पास डबल बॉन्ड हो तो आप ज्योमेट्रिकल आइसोमेरिज्म पॉसिबल कर सकते हो, E और Z कर सकते हो अकॉर्डिंग टू अरेंजमेंट ऑफ ग्रुप्स अराउंड द कार्बन कार्बन डबल बॉन्ड. सेकेंड, आप इसमें एक कायरल सेंटर भी देख रहे हो. So if you see in first case, your chiral center has a configuration R and your double bond has a configuration Z. Okay? अब ये आपने एक मॉलिक्यूल सिंपल लैब में आपको सिंथेसाइज करने के लिए दिया. दिस इज अ इंसेक्ट फेरेमोन. ओके, ये आपको सिंथेसाइज करने के लिए दिया, आपने बहुत एफिशिएंटली सिंथेसाइज किया. 99% आपने एसिमेट्रिक सिंथेसिस अचीव कर ली इसमें. Can you see presence of 1% of S and Z isomer can inhibit activity completely. So if a synthesis has been planned for such type of molecule, you are still getting the 99% one enantiomer in a pure form, the other 1% completely inhibit the activity of the 99%. So such is the requirement. Now the question come, why? How we people survive in COVID? Because our immune system responded differently. Some have not survived. Unfortunately, their immune system responded differently. But the virus is same. COVID virus is same for all. So this is the difference, that we don't know that how our immune system is working with respect to outer antigens. So if we are getting a drug which is 99% pure, my immune system is a very good one. So that 1% is not going to check, but someone is allergic to that thing. So presence of that 1% can lead to the drastic effect to that human being. So that's why if we are talking about the enantiomeric purity, if you check everybody, I pray that no one should take medicine, but if you have taken, if you check the back side of there is every percentage wise purity has been written, that is FDA or drug, गवर्नमेंट ऑफ इंडिया की जो ड्रग की इंस्टीट्यूशन है, दे अप्रूव इट ऑन द बेसिस ऑफ दैट परसेंटेज. Okay, so the requirement that comes is only with respect to this. Now I am just, you don't have to remember anything, you don't have to just see what is the mechanism, just see the change and the difference. Now in this case nothing is going to change in anything of the structure, atom, group, bonding, nothing, only the arrangement in 2 dimensions. So I am saying if you are drawing this molecule in your register, you are not going to see any change, but if you seeing in 3 dimension, just see the properties which they are showing. In first molecule, if you see, that is beta blocker for heart diseases, second is contraceptive. In below you see, one is no narcotic activity, the other is showing the narcotic activity. Now these molecules are different only in the arrangement. If you see the example of aspartame, you have a 4 different type of enantiomers and diastereomers which are possible, but out of this 4 only one is sweet. And the most latest example, that is of penicillin. The one molecule is antibiotic, the other molecule is inactive. Now can you explain me what is the difference between first and second? क्या difference आपको first and second में लग रहा है? Their phenyl, CH2, CO, NH bond, they are same, 4 member cycling ring. The only difference is, if you see carbon 3 and carbon 4, the presence of hydrogen atoms. In first case, if you see the arrangement of these 2 hydrogens with respect to each other, it is cis. Now in the next molecule, if you see the hydrogen, they are trans. Now can you see such a big molecule, only hydrogen atom which is just arranging whether it is on the same side and whether it is on the opposite side. It completely make the molecule active and inactive. So penicillin is the first antibiotic drug which nature has helped us to reach for that molecule. Now if you see the difference between penicillin and epipenicillin, it is just arrangement of hydrogens. Now if you see other chirality is also present, that is a bicyclic molecule, carbonyl group, amide group, all are present, but the difference is only single hydrogen atoms, whether they are cis or trans. That difference is only can lead to the formation of one is antibiotic and other is not antibiotic. So if you are planning to synthesize any such type of molecule and you are not seeing the configuration in a molecule, then you are able to synthesize a new molecule. You can do the molecular docking study, you can do the ADMI study, but when you try to put these molecules for biological activity, whether you are getting or not, it is a different aspect. Why? Because the appropriate configuration is required to hook with the molecules which are present in the living system. ये जो ड्रग मॉलिक्यूल है, जो सुबह उन्होंने आपसे ड्रग की डेफिनेशन पूछी थी, ये जो ड्रग मॉलिक्यूल्स है वो कहाँ यूज करने वाले हैं? वी आर नॉट पुट इन द ग्लास बॉटल एंड कीप इन द होम. उसको कोई सजा के रखने के लिए तो नहीं लाता है ना. दैट इज फॉर द लिविंग बीइंग. सो वो लिविंग बीइंग से कैसे रिएक्ट करेंगे, अगर उसकी थोड़ा सा भी बेसिस माइंड में नहीं रखा एंड जस्ट ट्राई टू प्रिपेयर अ मॉलिक्यूल, देन इट विल बी वेरी डिफरेंट एस्पेक्ट्स. ओके, so that is last one, that is we are starting asymmetric synthesis. Even when the other enantiomer is inert, it is still desirable to synthesize and use the active enantiomer only in pure form. So what I am talking about is asymmetric synthesis, that if you are getting more than one enantiomer, firstly you have to separate them, you have to purify them. Now it is a common practice that any new molecule, if it is in the market, you have to give the all the enantiomers, diastereomers, all the purity consent, then it can be put into the market. Okay, so we will continue tomorrow from here. Okay, sir, ठीक, बोलिए.
Thank you so much, sir, for this amazing lecture. Next up, I would like to request Professor Gurjasprit Singh to share a few words about our guest, Professor Narinder Singh.
Good afternoon to all of you. So, Professor Narinder Singh is a distinguished faculty member in the Department of Chemistry at the Indian Institute of Technology, IIT Ropar. He earned his PhD from Guru Nanak Dev University, Amritsar, and went on to pursue postdoc research in Japan, South Korea, UK, and before joining IIT Ropar in 2009. Professor Singh is known for his pioneer work in the field of supramolecular chemistry, material science, and chemical sensing. His research focuses on the design and development of fluorescent turn-on chemosensor, particularly using calyx for arenes, tripodal frameworks, ionic liquids, and nanoparticle-based system for the detection of environmentally and biologically relevant ions. He has significant contribution to lifestyle imaging, environment monitoring, and drug delivery research. With over 350 publications and more than 9,000 citations, he holds an impressive H-index of 45. His work has been featured in prestigious journals like JACS, JOC, Organic Letter, Talenta. Behind its research, he is a dedicated mentor to PhD scholars and actively involved in several national and interdisciplinary projects. His contributions reflect not only academic excellency but also a strong commitment to applied science for societal and strategic benefit. So now I invite Professor Ninder to deliver his talk. Thank you.
Yeah, thank you very much for the invitation. And my work, 50% work is concerned with the chemosensors and some other work for the applied application. Today, I am not going to talk about chemosensor. So my today's talk is about antimicrobial agent and their biomedical application and environmental application. Especially, we will talk about the surface engineering, means how to make the membranes, how to make the antimicrobial surfaces, how to make a self-cleaning surfaces. So those things we will discuss today. So those who are new to my presentation, so for them that I am mostly involved for the chemosensor and especially for it's not biosensor, only chemosensor for determine the toxin in soil and water. And once we know that okay, this industrial water is contaminated with one particular type of toxin, then we make the method for removal of those toxins. And that is also a water treatment. And some of the material which are the waste in the industry be converted into useful entities, and several of our technologies are being used in the industry. Today I will talk about the water treatment as well as self-cleaning surfaces. So if I talk about antimicrobial activities or self-cleaning surfaces, so AMR, first thing is AMR, that is antimicrobial resistance. What happen? Now, many people they are taking the drug molecule and some of the animal as well as poultry, every day they give small amount of antibiotic to those animals. And after that, those type of toxins, they go to the water bodies. And if there are n number of bacteria, and if you put first antimicrobial agent, it will kill some of the bacteria. But those bacteria which are resistant to that drug, then they will propagate like anything. Now, if the next drug will come, then out of those population, some will be killed and other will be resistant to the two drug. In this way, there is a generation of multi-drug resistant bacteria. So multi-drug resistant bacteria or AMR, antimicrobial resistance, it's a great concern of today's scientific community. And if we see that 4.95 million people in 2019, they were affected with this type of MDR bacteria. Even you may have realized that sometimes somebody is having skin infection, and after that you are treating with one antibiotic, another antibiotic, again you are not get rid of that type of skin infection, probably because of the resistant fungus is there in the body. Especially those type of things are happen in the toes of the forefoot. So those type of things are there. So some of the anti or bacterial disease include like pneumonia and other type of disease are there. So now if I see the history of antimicrobial drug, so first period was the pre-antibiotic era when the penicillin was invented. You know, for penicillin, Helmingen, he get the Nobel Prize, and after that there was a golden age when if there is any problem, they give the antibiotic agent, immediately they kill the material or kill the bacteria. But in the year of 1948, there was some of the bacteria which become resistant for the penicillin. Even there is another bacteria invented in India that is, in the penicillin we have lactam ring, right? We have a lactam ring. And in India, in New Delhi, and name is also New Delhi metallo beta lactamase enzyme. That enzyme is secreted by the bacteria. It will break the lactam ring. If it will break the lactam ring, it means penicillin or anything which is derived from the penicillin G, that will not come into the effect to the as far as antibiotic are concerned. So that's why again it will be a resistant variety. So after that there was a era when the things happen me too drug, means a large number of drugs were synthesized, not only this beta lactam based drug, there are large number of other drugs which were synthesized, and after that they realized that some of around 60 antibiotics were released into the market. Spalosporin is one, carbapenem is one. So those type of drugs are the example which was invented in this year. And after that, there is a discovery void, means if I see the literature between 2001 to today, there are hardly any drug which was coming to the market and which was invented and after that it is coming to the market. Most of the time repurposing of drug is happening, and we also never take that type of risk to make a new drug for the market. Actually, we have also combined some of the drugs using supramolecular interaction. Let us say if I have one drug, if I have two drugs, if I combine in such a way that after that they will not lose their chemical identity, but they become such a material that they will give a some synergetic effect, then it will be a reasonably good progress. So those type of things are going on at the moment, and I will discuss those things today also. So if I have to see the drug resistant bacteria MRSA, actually I have also worked for MRSA. In fact, we have invented some of the compounds which are active against the MRSA. That is S06, it becomes a resistant for S06, it's a resistant variety. If you see, India is also, although more than 80% means red color, orange color means around 60 to 70% area of India is also affected with this MRSA. And there is also another drug that is carbapenem. There are bacteria which are resistant to the carbapenem, and that is quite predominant in the Indian states. And there is another third generation resistant varieties are also there in the India. Even fluoroquinolones based drugs, they are also resistant in the India. So means there are large number of bacterial colonies which are there in the country, for which we have to address the problem by giving a new direction for the bacteria. If I see the literature, earlier literature, if you see, then at that time this beta lactam ring was there, be it is carbapenem, be it is phylosporin, and after that there is a generation of some of the antimicrobial peptides are there in the literature. Some people made self-penetrating peptide so that they will enter to the microbes, and after that they will cause some of the antimicrobial activities. And after that, with the peptides, there is one problem. One is the synthesis of peptide, that is a major problem. Another problem with peptide is you have to keep at a ambient temperature, and if there are unregulated amount of any salt, then peptide most of the time they will coagulate or they will lose their activity. So that's why after that those who are working in the area of organic chemistry, they thought, let us combine some of the features of those type of peptides in the form of peptidomimetic. So basically peptidomimetic means looking almost similar to the peptide, so they made the peptidomimetic like that way. And person who are belonging to my community means supramolecular scientist, we try to synthesize those type of molecules. So we were thinking what are the application or what are the affinities which we are getting from this antimicrobial agents. So basically it's a supramolecular assembly, means there is some are hydrogen bond donor, some are the hydrogen bond acceptor. So when this type of molecule are there in the literature, peptidomimetic based compound are there in the literature which are offering too much hydrogen bond donor site, too much hydrogen bond acceptor site, then the people like me, we thought, let us try our fate with the molecule who are good hydrogen bond donor, who are good hydrogen bond acceptor, and top on that, let us put some of the molecule like heterocyclic moiety. Let us see what is happening with this type of molecule. So that is another generation of material which are there in the literature. And top on that, some of the metal ion like silver, you know that it's a good antibacterial agent, and copper. During corona period, we have also invented some of the formulation for antibacterial activity using copper nanocluster, which were eventually patented, and that technology was transferred to the industry, and today it is commercially available in the market, which was invented in my lab. That was the copper nanocluster based things. So I mean to say that there are large number of strategies by which you can attain the antimicrobial properties out of the system. So how those antibiotics, how they are working? either first thing is either they will break the cell wall of bacteria, and if the cell wall will break down, then protoplasm will come outside from that bacteria. Another thing is there is a flux pump, means whatsoever waste material is there, bacteria get rid of that one. So some of the drug affect that pump. So that's why again toxin accumulate in the protoplasm of bacteria. And another is they are interacting with the cellular enzyme. Enzyme has to do some work, and if there are some of the drug molecule which interact with the those type of enzyme, and after that it is affecting the activity of enzyme, automatically it will disturb the normal functioning of bacteria, and some are inhibiting by the in the protein biosynthesis. So large number of mechanisms are there. So my method of working is like that way. At least you attack at least on two sides. Either I work with the membrane along with the inhibition of enzyme, or I will work with something it will combine with, or it will interact with the DNA, as well as it is affecting this flux pump. So I am try to combine two approaches together in my drug molecules. So we will discuss that later on. So now if we see peptides, linear peptide are there in the literature, which have shown excellent antibacterial activity, and some of the molecules which we have having that type of tryptophan like of moiety, that are also extensive in the literature along with the guanidine salt. And another is again peptide molecule. So if I see peptide or peptidomimetic type of thing, I have noticed one thing, that either that tryptophan like fluorophore or tryptophan is there, another is guanidine is there. So I thought, let us combine these structure feature in the new group of our compounds. So and first is what is the benefit of this type of peptide? If you ask me that, okay, there are large number of peptide, I have shown over here why peptide are so fascinating and what activity I will get out of these peptides. So activity I will get in that way. Either those peptide, let us say this is the cell membrane of that, let us say this is the bacteria and this is the cell wall of that bacteria. Either these peptide will create this type of barrel shape model, means it will create a hole and from where protoplasm will come out, or it will create the storipod pore, or carpet model, means this is the bacterial cell wall, something is attaching here, something attaching here, something here, something here. After that, whenever there will be bacterial fission from that area where we have that carpet model, the protoplasm will come out, and after that it will create the bacterial cell death. So although there are some of the limitation, we know peptide has limitation. First limitation is it's a peptide, it's a amide bond is there, and if there is a pepsin type of enzyme, tryptophan is there, those type of enzymes are there, immediately protein will degrade and peptide will degrade, and and after that if my peptide will degrade, then it means it will not give the activity whatsoever I am anticipating from that molecules. Another is cytotoxicity. Some of the peptide, they are cytotoxic, like snake venom. It's a one type of protein, but it is toxic. Lack of oral bioavailability, these things, this I will take little later. And resistance to the bacteria. Bacteria become very resistant to this type of peptide. There are mechanism, I will not go to that detail, and production cost is too much high. If I have to generate a peptide molecule having 4 or 5 amino acid and synthetically I have to commercialize it, it is very expensive business. So that is a big hurdle. And another problem is the stability of that formulation. As I said, that if there is a interaction of, if there is a high salt strength, if there is a alteration in the pH, immediately my peptide molecule will break down. So means stability of formulation is a big issue. To counter all these things, now before going to the design of my molecule, let me show you that most of the time if we have zinc oxide, we have silver, we have copper oxide, they are acting as a antimicrobial